Imagine standing at the threshold of medicine's grand cathedral. The vast doors swing open, and what greets you is not the glitter of cures or the clamor of surgical triumphs, but the quiet pulse of pathology, the science that dares to ask why disease begins, how it unfolds, and what it leaves in its wake. Pathology, dear listener, is the great interpreter of suffering. It is the discipline that listens to the whispers of damaged tissues, deciphers the cryptic language of mutated DNA, and reveals the hidden drama unfolding within the sanctum of our cells. If physiology is the symphony of life, pathology is the discordant note that transforms harmony into struggle. And yet, without pathology, medicine would stumble blind. It is here that the bridge between the serene world of the basic sciences and the urgent cries of clinical practice is constructed. Morphology, microbiology, immunology, molecular genetics all lend their voices, and pathology harmonizes them into an understanding of disease. Think of Rudolf Virchow, the father of modern pathology, who in the 19th century declared, "The seat of disease is the cell." With that proclamation, he shifted the lens of medicine from vague humors and spirits to the tangible world of cellular life. The dictum remains unshaken: when cells are sick, the body is sick. Thus begins our story. We leave behind the healthy landscapes of Chapter 1 and descend into the darker forest where cells face enemies: oxygen deprivation, chemical poisons, genetic flaws, infectious invaders, and the betrayal of immunity itself. This is the realm of cell injury, cell death, and the remarkable ways in which life resists and adapts. The core of pathology. Every disease, no matter how intricate, can be unraveled into 4 elemental threads. 1. Etiology, the cause. Sometimes obvious, like the sharp trauma of a blade, sometimes cryptic, like a single base mutation in a gene. 2. Pathogenesis, the unfolding tale of how cause begets consequence, the stepwise march of molecules, organelles, and tissues into dysfunction. 3. Morphologic changes, the visible footprints, be it the shrunken nucleus, the swollen mitochondrion, or the necrotic heart muscle pale against the magenta of the living. 4. Clinical manifestations, the symptoms and signs that bring a patient to the bedside, the fever, the pain, the breathlessness, all rooted in cellular catastrophe. It is a sequence, a chain, from the molecular spark to the structural collapse to the human experience of disease, and our task in this chapter is to trace that chain from its beginning. A dance between stress, adaptation, and death. Let us step closer. Picture a healthy cell, a small citadel, held steady by a delicate balance called homeostasis. Its metabolic state, its specializations, and its dialogue with neighboring cells all keep it within narrow bounds. But life is seldom merciful. A surge of blood pressure, a sudden infection, a toxin, a fever, a demand for more than it can give, these stressors knock upon the cell's gates. What happens then? Cells are not helpless. They adapt. They enlarge in size, hypertrophy, to bear the load. They increase in number, hyperplasia, to share the burden. They shrink, atrophy, to conserve precious resources. Or like actors in a masquerade, they even change their identity, metaplasia, to better suit hostile conditions. Such adaptations are reversible, pragmatic, survival strategies. But every adaptation has a price. Push too far, and the citadel begins to crack. If the stress continues, If the insult is unrelenting, adaptation collapses into cell injury. First, reversible. The cell swells with water. Its endoplasmic reticulum dilates. Ribosomes detach. Chromatin clumps. It staggers, but may yet recover. Remove the stress, and life resumes its course. But there is a point of no return. Mitochondria fail, membranes rupture, lysosomal enzymes spill like acid upon fragile machinery. The injury becomes irreversible, and the cell marches inexorably toward death. The two roads to death. When cells die, they do so upon two main stages. One is necrosis, a messy, catastrophic end. Membranes burst, organelles disintegrate, enzymes digest the corpse, and the cell's contents spill into the surrounding tissue. The neighborhood responds with inflammation, as though cleaning the aftermath of a violent accident. The other is apoptosis, a death with elegance, with choreography. The cell activates its own suicide program, packages itself neatly into apoptotic bodies, and signals phagocytes to carry away the remains, silently, without inflammation, without fuss. Beyond these two lie other, stranger deaths: necroptosis, pyroptosis, pharoptosis, and the strange act of self-cannibalism called autophagy. We will meet them all in due course. The larger picture. But death is not the end of our tale. From injury and death emerge other phenomena. Cells may hoard lipids, proteins, pigments, or glycogen, cluttering their cytoplasm with unwanted treasures. And calcium, once carefully balanced, may calcify dead tissues or settle in living ones. And as time marches forward, aging etches its marks on every cell. Telomeres shortening, DNA damage accumulating, repair mechanisms faltering. Thus, the story of cell injury is not merely a catalogue of destruction. It is the very foundation of pathology, the soil from which every disease grows. So far, we have opened the door to pathology, traced its four cardinal threads, and glimpsed how cells respond to stress with adaptation, injury, or death. The stage is set. Up next, we descend deeper into specifics, the causes of cell injury, where we will meet oxygen starvation, chemical poisons, infectious invaders, and the treachery of our own immune system. Each one leaves its unique signature upon the vulnerable cell.
Section 2. Causes of Cell Injury. Let us now turn our gaze upon the question that has tormented physicians for centuries: what brings ruin upon the cell? If life at the microscopic level is so delicately balanced, then it is equally true that 1,000 enemies conspire to disturb it. The causes of cell injury are as varied as the human experience itself, ranging from the obvious brutality of a car crash to the silent treachery of a gene that falters in its duty. Above all, there is one enemy that reigns supreme: oxygen deprivation. Without oxygen, the machinery of respiration collapses. Picture the mitochondria, those powerhouses humming in their orchestral rhythm, suddenly silenced. ATP dwindles, ion pumps falter, membranes swell, and the citadel of the cell is left gasping. This condition we call hypoxia. The causes of hypoxia are legion. Sometimes the blood itself is starved, as in shock or massive blood loss. Sometimes the airways falter, suffocating lungs unable to draw in breath. Sometimes the hemoglobin that should carry oxygen is poisoned by carbon monoxide, which binds with a passion greater than oxygen itself, or by profound anemia that strips the blood of its couriers. And perhaps most dangerously, sometimes the blood flows no more, as in ischemia, where a vessel narrows or clots and starves its downstream territory. Whether the result is a silent atrophy or a thunderclap of infarction, the end is the same. The cell trembles, falters, and may die. Yet hypoxia is only the first of many foes. The world is full of physical agents that strike the cell without subtlety. A sudden blow from trauma, a burn that sears proteins beyond recognition, a frost that freezes membranes stiff, or the crushing weight of atmospheric shifts. These are assaults nature hurls against the body. Add to these the silent arrows of radiation, both ionizing and ultraviolet, and the invisible lash of electrical currents, and one begins to see the universe as an arena constantly testing the resilience of life. Then come the chemicals, more insidious, for they wear many masks. Some are simple, an excess of salt, an overdose of glucose, seemingly benign molecules that, in overwhelming concentration, become toxic. Others are infamous poisons, arsenic, cyanide, mercury, small in dose, catastrophic in effect. But the true irony lies in how even our companions become enemies. Oxygen itself in excess becomes destructive. The medicines designed to heal carry side effects that scar tissues. Alcohol, inhaled pollutants, industrial solvents, insecticides, all in their own measure, gnaw at the cell's defenses. The modern age, for all its advances, surrounds us with a chemical storm that our cells must weather. But sometimes the invader is alive. Infectious agents form one of the most ancient and persistent scourges. Imagine the spectrum, viruses smaller than the eye of a needle, weaving their genomes into ours, bacteria multiplying with ruthless efficiency, fungi creeping through tissue, parasites coiling in the gut or swimming in the blood. From the delicate rickettsia to the monstrous tapeworm, life attacks life, and the cell pays the price. Each microbe devises its own strategy. Some produce toxins, others tear membranes, still others hijack cellular machinery until the host itself becomes a factory of its destroyer. And then, perhaps most tragically, the cell is betrayed by its own guardian. The immune system, meant to shield, may sometimes turn its sword upon the self. In autoimmune disease, the body mistakes its own tissues for invaders, launching volleys of inflammation upon the innocent. At other times, an exaggerated immune reaction to harmless substances, like pollen, dust, or certain foods, produces collateral damage that far exceeds the original threat. The paradox is bitter. The very system that saves us daily from microbial death may also carve wounds into our tissues. The list of culprits does not end there. Hidden in the spiral of DNA are genetic abnormalities that can condemn cells before they are even born. Some are gross errors, an extra chromosome as in Down syndrome. Others are minute, a single base substitution that reshapes hemoglobin into the sickle's blade. Genes may code for defective enzymes, leaving entire metabolic pathways crippled, or allow the accumulation of misfolded proteins, each a tiny time bomb within the cytoplasm. Even common variations in DNA, those so-called polymorphisms, may tilt the balance between resilience and vulnerability when faced with environmental stress. And finally, there is the great irony of nutrition. For want of it, millions perish. Children with swollen bellies, their bodies wasting from protein-calorie malnutrition, the hollow eyes of starvation etched in human history. Vitamins, too, in their absence bring devastation. Rickets, scurvy, pellagra, beriberi. Yet in other corners of the globe, it is the abundance that kills. Obesity marches in hand with diabetes, hypertension, and cancer. A diet laden with lipids and cholesterol clog arteries and set the stage for atherosclerosis, the silent killer of the modern age. So both famine and feast conspire against the fragile equilibrium of the cell. So whether it be the choking absence of oxygen, the brute force of trauma, the venom of chemicals, the cunning of microbes, the betrayal of immunity, the ticking clock of defective genes, or the imbalance of diet, every road of injury leads inexorably to the same battlefield, the vulnerable cell. And here, one truth emerges. The cell is not destroyed in a single blow. There is always a progression, a journey from the subtle to the catastrophic. First reversible, then irreversible, finally death. This journey, the progression of cell injury and its march toward demise, is where we must venture next.
Section 3. The progression of cell injury and death. A cell's journey into injury is not a sudden plunge into darkness. It is a sequence, a slow unraveling, almost like a tragic play in acts. The curtain rises with the faintest of changes, subtle biochemical murmurs that no naked eye could perceive. These earliest shifts are whispers, detectable only with sensitive tools, enzymes altering their pace, membranes flickering with ion imbalance, chromatin beginning to clump. Hours may pass before these invisible changes leave visible scars upon the tissue. Imagine a heart deprived of blood by a blocked coronary artery. Within mere minutes, the muscle cells swell, their pumps failing for lack of ATP. Yet at this point, if the blood flow is restored, the cells may recover, as though shaken but not broken. But linger longer, let the deprivation persist, and the cells cross a threshold, an invisible line known grimly as the point of no return. Beyond it, even if oxygen surges back, the mitochondria cannot rouse themselves. Membranes rupture, lysosomes burst, the end has been written. Thus, the progression of cell injury can be envisioned as a spectrum, from the mild, reversible disturbances to the grave, irreversible collapse that culminates in death. In reversible injury, two features stand out. First, swelling. The cell, robbed of its ability to control ion balance, allows water to rush in. Organelles balloon, the endoplasmic reticulum dilates, ribosomes detach, protein synthesis stutters. On the microscope slide, the cytoplasm appears vacuolated, as though filled with tiny clear bubbles, hydropic change or vacuolar degeneration. It is the earliest hallmark, a cry for help. The second feature is a change in the cell's very hue. Stain these injured cells with hematoxylin and eosin, and they glow with a deeper red, a pronounced eosinophilia. This happens because RNA, normally binding the blue hematoxylin, dwindles. Proteins, denatured and unfolded, hungrily absorb eosin's scarlet. The color is a foretelling. The cell is on the path toward death. Ultrastructurally, the tail grows more vivid. The plasma membrane forms blebs, delicate protrusions that herald weakness. Microvilli, once abundant, vanish. Mitochondria swell and sprout amorphous densities. Myelin figures, whorls of lipid from fractured membranes, appear like funeral garlands drifting in the cytoplasm. Even the nucleus shows its distress, chromatin clumping as though drawing into itself for protection. And yet, at this stage, hope remains. Remove the insult, restore the balance, and the cell may return to its former state, carrying no permanent scar. But should the injury persist, a darker transformation unfolds. The cell steps into irreversible injury. Two events announce this fatal transition. Mitochondria, those vital furnaces, suffer irretrievable dysfunction, unable to generate ATP, even if oxygen returns. And the cell's membranes, both plasma and lysosomal, surrender. With their rupture, the cell dissolves from within, its own enzymes unleashed like wolves in a collapsing citadel. Here lies the path to cell death, which can manifest in different guises. The chaotic, inflammatory demise of necrosis, the deliberate, clean execution of apoptosis, or other less common forms that blur the boundaries. Regardless of the path, the destination is the same, the end of cellular life. Consider again our patient with a myocardial infarction.
In the first hour, swelling and reversible injury reign. If blood flow is restored, recovery is possible. But let the occlusion linger for 4, 6, 12 hours, and the muscle cells will die irrevocably. Under the microscope, nuclei vanish, cytoplasm fragments, and pale zones of necrosis stand stark against the surrounding magenta tissue. In the blood, proteins like troponins escape from ruptured membranes, rising as early heralds of irreversible death. Thus, we see the drama of cell injury as a continuum, a tragic arc. Adaptation, reversible injury, the crossing of the fatal threshold, and finally, cell death. But death itself is not a singular fate. It wears many masks, and it is here, at this pivotal juncture, that we must linger. For the next chapter of our journey unveils the two principal phases of cellular demise, the violent disintegration of necrosis and the choreographed silence of apoptosis.
Section 4. Cell death, necrosis and apoptosis.
When a cell dies, it does not simply vanish. It leaves behind a story written in its morphology, a testimony visible under the microscope, and sometimes a trail of signals detectable in the bloodstream. To the pathologist, these clues are the footprints of the cell's final journey. The most ancient and perhaps most brutal of these endings is necrosis. Picture a battlefield after an explosion, membranes torn asunder, organelles shredded, cytoplasmic contents spilling into the extracellular world. This is necrosis. Disordered, uncontrolled, inflammatory. The immune system sees the spillage as danger and summons its forces. Neutrophils flood in, macrophages follow, and the tissue becomes a theater of inflammation. Necrosis is the cell's scream, its messy end broadcast to the surrounding world. Microscopically, necrosis carries distinctive signatures. The cytoplasm grows glassy and more eosinophilic, proteins denatured and bound irrevocably to dyes. Vacuoles may appear, organelles fragment, and calcification may sprinkle over the remains, like dust upon ruins. The nucleus, once the proud custodian of the genome, undergoes a trilogy of death: first condensation, then fragmentation, and finally dissolution into nothingness. But necrosis is not uniform. It paints different canvases in different tissues, and so pathologists classify its patterns. Coagulative necrosis, the hallmark of ischemic injury in solid organs like heart, kidney, and spleen. Here, the basic outlines of the cell persist like ghostly silhouettes, even as nuclei vanish. Infarcts in these organs stand firm and pale, as if chyme has frozen the architecture. Liquefactive necrosis, in contrast, is dissolution incarnate. Seen in bacterial infections and in brain infarcts, enzymes digest tissues into liquid viscous mass. In the brain, a stroke may leave behind a soft, cystic cavity, a cavity of absence. Caseous necrosis, the archetype of tuberculosis, looks crumbly and white, reminiscent of cottage cheese. Microscopically, it is an amorphous collection of debris surrounded by a border of granulomatous inflammation. Fat necrosis is not a distinct mechanism, but rather a descriptive phenomenon, classically seen in pancreatitis. Pancreatic lipases escape, splitting triglycerides into free fatty acids, which then bind calcium to form chalky white deposits, soap-like, gritty to touch. Fibrinoid necrosis, subtle and rarefied, occurs within immune-mediated vascular damage. Immune complexes and plasma proteins deposit within vessel walls, staining them bright and eosinophilic, like ribbons of fibrin under the microscope. Each of these patterns is a dialect of the same language, the language of irretrievable cell death. But necrosis is not the only mode. There is also apoptosis, the quiet alternative, the cell's dignified suicide. Where necrosis shouts, apoptosis whispers. It is a process written into the very script of development and homeostasis. In the embryo, apoptosis sculpts fingers by removing the tissue between them. In adults, it prunes excess cells, eliminates self-reactive lymphocytes, and disposes of damaged cells without raising the alarm of inflammation. Morphologically, apoptosis is delicate and precise. The cell shrinks rather than swells. Its chromatin condenses into dark crescents. Its cytoplasm buds into neat apoptotic bodies, each wrapped in membrane. These fragments display signals, like phosphatidylserine flipped to the outer leaflet, that invite macrophages to engulf them. The corpse is cleared silently, without leakage, without inflammation, as though the cell had bowed and exited the stage without disturbing the performance. The biochemistry of apoptosis reveals two great pathways, both converging upon a common execution. The intrinsic, or mitochondrial pathway, begins when the balance of life and death proteins within the BCL2 family tips toward pro-death members like BAX and BAC. Mitochondrial membranes become permeable, cytochrome C is released into the cytoplasm, and a caspase cascade is ignited. These caspases are the executioners, proteases that dismantle the cell with surgical precision. The extrinsic, or death receptor pathway, is triggered from within. Signals such as Fas ligand or tumor necrosis factor bind to their receptors, assembling a death-inducing signaling complex that activates caspases from the cell surface inward.
Both routes converge upon the executioner caspases, which cleave vital substrates, dismantle the cytoskeleton, and fragment the nucleus. The result is a cell cut apart by its own hands, neatly packaged for disposal. Apoptosis is seen not only in physiology but also in pathology, in viral infections where cytotoxic T cells induce suicide of infected targets, in DNA damage that overwhelms repair, in cancers treated with chemotherapy, where tumor cells are coaxed into self-destruction. It is the body's way of sacrificing the few for the survival of the many. So, side by side, necrosis and apoptosis reveal two contrasting philosophies of death. Necrosis is chaotic, accidental, inflammatory. Apoptosis is ordered, intentional, silent. One provokes a riot, the other a whisper. Both, however, remind us that cellular mortality is an inseparable part of life's grand design. And yet, the story does not end with these two. Modern science has unveiled other modes of demise, stranger, hybrid forms that blur the old distinctions. Necropoptosis, pyropoptosis, pheropoptosis. Unconventional deaths that we must now explore, for they widen the very definition of what it means for a cell to perish. Section 5. Unconventional forms of cell death: necroptosis, pyroptosis, pheroptosis, and autophagy. Necrosis and apoptosis once seemed to span the entire spectrum of death. But in the last few decades, as microscopes grew sharper and molecular biology unraveled intracellular dialogues, other faces of mortality emerged. They do not conform neatly to old categories. They are hybrids, part chaos, part order, bearing features of both necrosis and apoptosis, yet belonging fully to neither. One such form is necroptosis. The very word is a marriage: necrosis in its morphology, apoptosis in its regulated mechanism. Picture a cell that has chosen suicide but executes it with the messiness of an explosion. Unlike apoptosis, necroptosis does not rely on caspases. Instead, it is orchestrated by kinases with peculiar names, RIPK1 and RIPK3. Serine-threonine kinases that bind and form a deadly complex. This complex unleashes signals that perforate membranes, deplete ATP, and provoke the swelling and bursting of the cell.
In appearance, necroptosis is indistinguishable from necrosis: ruptured membranes, inflammation, leakage. Yet beneath the chaos lies a molecular program, written and controlled. Necroptosis has clinical resonance. It is seen in certain viral infections where viruses sabotage the caspase machinery, forcing the host cell to choose another mode of death. It is implicated in neurodegenerative diseases, in ischemic injury of the brain and heart, in inflammatory disorders where the storm of cytokines triggers this pathway. Necroptosis is the cell's paradoxical option. If you block one death road, it takes another, equally destructive.
If necropoptosis is death by bursting, pyropoptosis is death by fire. This is the mode of cells that fall victim to microbial invasion, particularly those ambushed by bacteria. Pyropoptosis is mediated by a multi-protein sentry known as the inflammasome. When pathogens or danger signals awaken this sentinel, it calls upon an unusual executioner, caspase 1, sometimes caspase 11 or 4 and 5 in humans. These caspases do not dismantle the cell quietly. Instead, they cleave gastromin, a protein that inserts into membranes to form fiery pores. The cell swells, ruptures, and spews its contents, especially the potent cytokine interleukin-1 beta and interleukin-18. The tissue responds with fever, pain, and a storm of inflammation. Pyropoptosis, then, is not just a death, but an alarm bell. It sacrifices the infected cell to warn the organism. It is the equivalent of a burning watchtower collapsing while sounding the alarm to rouse the immune army. Such is its role in bacterial infections, sepsis, and even sterile inflammation triggered by crystals or metabolic byproducts. Another novel pathway is ferropoptosis, an iron-dependent form of death. This is no quiet process, but a catastrophic chain reaction of lipid peroxidation. Imagine the cell's membrane lipids as a library of fragile manuscripts. In ferropoptosis, free iron acts as a mischievous arsonist, catalyzing the formation of free radicals that ignite the lipids into peroxidation. One by one, the pages catch fire, and the membrane integrity collapses. Unlike apoptosis, there is no chromatin condensation, no neat apoptotic bodies, only oxidative devastation of membranes. Ferropoptosis has captured attention in cancer research. Many tumors rich in iron and with altered metabolism are vulnerable to it. Drugs that promote ferropoptosis may yet prove to be weapons against cancers resistant to traditional therapies. It is also implicated in neurodegenerative diseases like Parkinson's and Alzheimer's, where iron accumulation and oxidative stress conspire against neurons. And then comes a process not strictly death, but a strange in-between, autophagy, literally self-eating. Autophagy is, in its essence, a survival tactic. When nutrients are scarce, the cell forms double membrane vacuoles, autophagosomes, that engulf damaged organelles or proteins. These fuse with lysosomes, and the cargo is digested, recycled into energy and building blocks. It is as though the cell, trapped in famine, consumes its own furniture to keep the hearth burning. Autophagy is not merely a response to starvation. It is also a housekeeping function, clearing away defective mitochondria, misfolded proteins, and aggregates that would otherwise poison the cell. In doing so, it preserves cellular integrity and prevents disease. Yet, when excessive or dysregulated, autophagy tips into self-destruction, dismantling essential components until the cell cannot recover. Pathologically, autophagy plays roles in cancers, infectious diseases, neurodegeneration, and inflammatory conditions. Some microbes, ironically, exploit it, using the autophagic vesicles as hideouts. Others are destroyed by it. Autophagy, then, is a double-edged sword, part guardian, part executioner.
Thus, we see that the catalog of cellular death is richer and more complex than we once believed. Necropoptosis blurs the line between order and chaos. Pyropoptosis turns the cell into a flaming beacon of alarm. Ferropoptosis burns membranes with the chemistry of iron. Autophagy hovers between life and death, a feast upon the self. Together, they remind us that death is not monolithic, but manifold, shaped by context, by insult, by evolutionary design. The cell, when pressed to the limits of endurance, has more than one script to follow in its exit. And so, having explored these newer roads to mortality, we are ready to return to the bedrock of our understanding, the mechanisms of cell injury. For whether by necrosis, apoptosis, or these alternative fates, the common thread is that cellular machinery fails in fundamental ways. Let us next unravel those hidden mechanisms.
Section 6. Mechanisms of cell injury. Every form of injury we have witnessed, whether oxygen deprivation, toxins, radiation, infection, or immune attack, converges upon a limited number of final common pathways. It is as though the insults are many, but the doors through which they enter are few. If we wish to master pathology, we must understand these doors, these critical mechanisms. The first and most fundamental is the depletion of ATP, the very currency of life. Cells are powered by adenosine triphosphate, generated chiefly within mitochondria through oxidative phosphorylation. Imagine a bustling city suddenly stripped of its electricity. Traffic lights fail, pumps stall, water stops flowing, communications collapse. So too does the cell when ATP dwindles. Sodium-potassium pumps cease, sodium and water rush in, the cell swells, organelles balloon. The endoplasmic reticulum dilates, ribosomes detach, protein synthesis halts. With prolonged depletion, the cytoskeleton collapses and the membranes become porous. Even glycolysis, hastily upregulated, provides little respite, leading to lactic acid accumulation and acidosis. The pH falls, enzymes falter, chromatin clumps. A cascade is set into motion, and if mitochondria cannot be revived, the march toward necrosis is irreversible.
But mitochondria themselves are more than powerhouses. They are arbiters of life and death. When their membranes are damaged, they do not simply fail in energy production, they actively release pro-death signals. Cytochrome C, once a loyal participant in electron transport, becomes the herald of apoptosis, spilling into the cytosol to activate caspases. In this way, mitochondria embody a paradox, guardians of vitality in one breath, initiators of suicide in the next. Another recurrent theme in cell injury is the influx of calcium. In the healthy state, calcium is carefully sequestered within mitochondria and endoplasmic reticulum, kept at vanishingly low concentrations in the cytosol. But when membranes falter or pumps fail, calcium floods in. This innocent ion then becomes an executioner, activating enzymes that degrade proteins, lipids, and nucleic acids. Proteases chew the cytoskeleton to tatters. Phospholipases gnaw at membranes. Endonucleases cleave DNA into fragments. At the same time, excess calcium triggers the opening of mitochondrial permeability transition pores, collapsing oxidative phosphorylation. Thus, the very element that normally signals contraction in muscle or secretion in glands turns into an agent of dissolution. Linked to these failures is the destructive force of oxidative stress. Oxygen, while essential for life, carries within it a hidden danger, the potential to form free radicals, reactive oxygen species with unpaired electrons seeking havoc, superoxide anion, hydrogen peroxide, hydroxyl radical. These are the marauders of biochemistry. Generated in mitochondria by radiation, by drugs, or by inflammation, they set upon lipids, proteins, and DNA with merciless efficiency. Consider lipid peroxidation. ROS attack polyunsaturated fatty acids in membranes, generating peroxides that propagate further radical reactions, like fire leaping from rooftop to rooftop in a crowded city. Membranes lose integrity. Ion gradients collapse. Proteins, too, are oxidized, their sulfahydryl groups cross-linked, their enzymatic activity crippled. DNA suffers breaks, bases are modified, mutations arise. Left unchecked, oxidative stress can drive a cell into death and a tissue into disease. And yet, cells are not defenseless. They harbor antioxidants, vitamins E and A, glutathione, enzymes like catalase, superoxide dismutase, glutathione peroxidase. These stand as firefighters, quenching radicals before they spread. But when the insult overwhelms defenses, the balance tips toward destruction. Another key mechanism lies in membrane damage itself. Once the plasma membrane is disrupted, the cell loses control over its internal environment. Swelling worsens, contents leak out, and inflammatory mediators are released. Lysosomal membranes may rupture as well, spilling hydrolytic enzymes into the cytoplasm. Nucleases, proteases, glycosidases that digest the very cell that housed them. This autodigestion transforms reversible injury into irreversible catastrophe. Beyond mitochondria, calcium, and oxidative stress, we must note the role of endoplasmic reticulum stress. The ER is the cell's factory, folding proteins with exquisite precision. But when misfolded proteins accumulate, because of genetic mutations, ischemia, infection, or toxins, the ER signals distress. It halts protein synthesis, upregulates chaperones to refold proteins, and attempts repair. Yet if the load is too great, if the unfolded protein response fails, the ER activates apoptosis. Thus, diseases as diverse as neurodegeneration, diabetes, and cancer find part of their roots in this misfolded burden. To these mechanisms, we may add DNA damage responses, inflammation-mediated injury, and defects in mitochondrial dynamics, each a page in the complex manual of cellular destruction. But the essence is clear. Whether by ATP depletion, mitochondrial dysfunction, calcium influx, oxidative stress, membrane rupture, or ER stress, all roads lead to the same outcome: collapse of homeostasis, loss of function, and cellular death. Understanding these pathways gives pathology its predictive power. It tells us not only why a cell has died, but how we might intervene. Could antioxidants protect? Could calcium channel blockers stave off injury? Could modulating apoptosis alter disease? These questions animate modern therapeutics. And so, the hidden machinery revealed, we now turn to see these mechanisms in action in the clinic. For no knowledge is complete until we witness how hypoxia, ischemia, reperfusion injury, and chemical poisons write these molecular failures into the living canvas of disease.
Section 7. Clinico-pathologic correlations: hypoxia, ischemia, reperfusion injury, and chemical injury. Of all the insults that afflict human cells, oxygen deprivation is the most universal and the most deadly. Life itself is but a delicate chain of oxidative reactions, and when oxygen is denied, the chain snaps. Hypoxia may creep in slowly, as in chronic respiratory disease, or it may strike suddenly, as when an embolus lodges in a vital artery. Its most dangerous form is ischemia, the loss of blood supply. For without blood, there is not only deprivation of oxygen, but also starvation of nutrients, accumulation of waste, and collapse of ionic balance. Consider the heart. Within minutes of ischemia, myocardial fibers swell, contractile function falters, and electric rhythms stumble. At first, the damage is reversible, the cells like drowning men who can still be pulled ashore. But prolong the insult, and the mitochondria fall silent, the membranes rupture, and necrosis unfolds. The patient clutches his chest in an infarction whose microscopic echoes we have already traced. The brain tells a parallel story. Neurons, exquisitely sensitive, die within minutes of ischemia, their loss permanent and devastating. Thus, oxygen and blood are not luxuries, they are the essence of cellular survival. And yet, an irony awaits. If the blood flow is restored after a prolonged ischemia, one might think recovery assured. But in truth, this sudden restoration can itself unleash further harm. This is reperfusion injury. When oxygen surges back into previously ischemic tissue, it feeds the mitochondria, yes, but also fuels the unchecked generation of reactive oxygen species. Free radicals pour forth like sparks in a dry forest, igniting lipid peroxidation and DNA damage. At the same time, the returning blood carries leukocytes that adhere to endothelium, migrate into tissue, and release proteases and oxidants. The complement system may also activate, compounding the attack. Thus, the very act of rescue may deepen the wound, as surgeons and cardiologists know all too well in the aftermath of thrombolysis or transplantation. Then there are the assaults of chemical injury, another vast category where toxins lay siege to the cell. Some chemicals act directly. Mercuric chloride, for example, binds sulfhydryl groups on membrane proteins, disrupting ion transport and increasing permeability. Cyanide paralyzes cytochrome oxidase, extinguishing oxidative phosphorylation in a heartbeat. These poisons require no transformation. They strike as they are, quick and ruthless. Others, however, are subtler, demanding metabolic activation within the body before revealing their toxicity. The liver, our great detoxifier, often becomes their stage. Acetaminophen, ingested in high doses, is metabolized into reactive intermediates that exhaust glutathione reserves, leading to lethal oxidative stress. Carbon tetrachloride, once used in industry, is converted into a free radical that initiates lipid peroxidation, swelling the endoplasmic reticulum, detaching ribosomes, and halting protein synthesis. The result is fatty change of the liver, progressing to necrosis. Even alcohol, friend to conviviality, betrays the liver when metabolized into acetaldehyde, disturbing lipid metabolism, generating ROS, and promoting fibrosis. Infections, too, add their contribution, for many microbes generate toxins or metabolic derangements that parallel chemical injury. Thus, whether ingested, inhaled, injected, or endogenously produced, chemicals test the resilience of cellular machinery. Some wounds are acute, others chronic, but all exploit the common pathways of ATP depletion, ROS generation, calcium influx, and membrane damage. So, in the clinic, cell injury manifests not as abstract pathways, but as infarcts of the heart and brain, reperfusion damage after stroke or surgery, hepatotoxicity from drugs and alcohol, pulmonary injury from pollutants, renal necrosis from poisons. The doctor at the bedside, when confronted with a failing organ, is in truth witnessing the drama of hypoxia, ischemia, reperfusion, and toxins playing out in the cellular theater. It is a reminder that the smallest units of life dictate the grandest outcomes of disease. And yet cells are not passive victims. They adapt. They change their size, their number, their function in remarkable ways to survive. To this resilience, to the story of adaptations of cellular growth and differentiation, we must now turn.
Section 8. Adaptations of cellular growth and differentiation. If injury represents the failure of equilibrium, then adaptation is its preservation by transformation. Cells are not rigid structures, they are dynamic actors, capable of changing their size, number, phenotype, or metabolic activity in order to meet new demands. These adjustments are not random, they are programs written deep into evolutionary history, allowing organisms to survive shifting burdens. Yet they are double-edged, protective at first, but sometimes laying the groundwork for disease. The first of these strategies is hypertrophy, the increase in cell size. Unlike hyperplasia, hypertrophy does not create new cells, it enlarges existing ones, swelling their cytoplasm and organelles to enhance function. It occurs when tissues face greater demands but lack the ability to divide. Skeletal muscle hypertrophies when weightlifters challenge it with resistance. Each fiber thickens with more myofibrils, mitochondria, and enzymes. The heart, when confronted with chronic hypertension or valvular disease, hypertrophies as well. Myocytes, incapable of division, grow larger to generate stronger contractions. Initially, this adaptation sustains the circulation, but with time, the overgrown myocardium outstrips its own blood supply, its mitochondria falter, and heart failure ensues. Thus, hypertrophy is at once a marvel and a menace. The second adaptation is hyperplasia, an increase in cell number. Unlike hypertrophy, it requires cells capable of proliferation. The driving force is growth factors, hormones, or signals that recruit resting cells into the cell cycle. Hyperplasia can be physiologic. The female breast during puberty and pregnancy expands its glandular epithelium under hormonal guidance. The liver regenerates astonishingly after partial resection, restoring mass by hepatocyte division. But hyperplasia can also be pathologic. Excessive estrogen may provoke endometrial hyperplasia, with abnormal bleeding as its clinical echo. Human papillomavirus infection induces growth factors that drive epithelial hyperplasia, producing warts. In such settings, the increased cell proliferation may provide fertile ground for mutations and progression to cancer. Thus, hyperplasia straddles the line between normal adaptation and precancerous risk. Next comes atrophy, the shrinking away of cellular mass. This is not simply death, but a downsizing, a retreat into minimalism. Cells reduce their size and metabolic activity, shedding organelles and cytoplasm. Atrophy may be physiologic, the uterus after childbirth, the embryonic notochord and thymus as development advances. Or it may be pathologic, skeletal muscle wasting after denervation, brain shrinkage in atherosclerosis, the emaciation of starvation. Mechanistically, atrophy combines decreased protein synthesis with increased protein degradation through the ubiquitin proteasome pathway. Autophagy joins the process, digesting organelles to recycle substrates. Under the microscope, autophagic vacuoles appear, sometimes containing remnants of mitochondria or ER. The ultimate result is a smaller, weaker, but still living cell, adapted to survival under reduced demand or supply.
And then there is metaplasia, the substitution of one differentiated cell type for another better suited to endure stress. It is a reprogramming orchestrated not by direct transdifferentiation, but by signals that redirect stem cells along new lineages. In the respiratory tract of a smoker, the normal ciliated columnar epithelium transforms into stratified squamous epithelium, sturdier against smoke's insults but bereft of ciliary clearance. In Barrett's esophagus, chronic acid reflux induces squamous epithelium to become columnar with goblet cells, better adapted to acid but vulnerable to malignant transformation. Thus, metaplasia illustrates the paradox of adaptation. It provides protection in the moment, but often at the cost of function and heightened risk of neoplasia. All these adaptations,
Hypertrophy, hyperplasia, atrophy, metaplasia are part of the same narrative. They reveal the plasticity of life, the ability of cells to mold themselves under pressure. But they also remind us of limits. Beyond a certain point, adaptation fails, mechanisms collapse, and injury ensues. And so we see that adaptation is not the opposite of injury, but rather its companion, a spectrum where growth, shrinkage, and transformation precede, accompany, or ward off damage. The cell in adapting whispers, "I will endure, but only for a while." And so, from the drama of adaptation, our gaze now turns inward into the secret vaults of the cell to explore the strange and telling world of intracellular accumulations and pathologic calcification.
Section 9. Intracellular accumulations and pathologic calcification. A cell is not merely a machine of metabolism, it is also a chamber of storage. Within its cytoplasm, substances are manufactured, trafficked, and disposed of with astonishing efficiency. But under certain conditions, this choreography falters, and the cell begins to accumulate material in abnormal quantities. Some of these are innocuous, others toxic, but all bear witness to disturbed homeostasis. The principles behind these intracellular accumulations are fourfold. First, a normal substance may be produced in excess or not adequately removed. Think of triglycerides in the liver during fatty change, where the balance between production and utilization tips toward storage. Second, an abnormal substance may accumulate, the result of defective folding or transport. Misfolded proteins, for instance, congregate as hyaline inclusions. Third, a normal endogenous substance may build up because of missing enzymes, as in genetic storage diseases. Glycogen, sphingolipids, or mucopolysaccharides swell within lysosomes, filling the cytoplasm with their burden. And fourth, an abnormal exogenous material may be deposited, as with carbon particles inhaled from polluted air or tattoo pigments pricked into skin. Among these accumulations, lipids stand prominent. The most common manifestation is hepatic steatosis, or fatty change of the liver. Alcohol abuse, diabetes mellitus, obesity, toxins, all can disturb lipid metabolism, leading to droplets of triglyceride filling hepatocytes. The organ appears yellow and greasy, and under the microscope, vacuoles push aside the nucleus in a striking pattern. Fatty change can be reversible, but unchecked, it may herald inflammation and cirrhosis. In macrophages, too, lipid accumulation appears in atherosclerosis, where cholesterol-laden foam cells populate arterial plaques, sowing the seeds of vascular disease. Next are proteins. In the kidney, heavy proteinuria leads proximal tubular cells to reabsorb proteins, forming reabsorption droplets. In plasma cells, immunoglobulins may crowd into Russell bodies. Misfolded proteins, unable to be degraded, stack into eosinophilic aggregates. The quintessential example is amyloidosis, where abnormal proteins form fibrils deposited extracellularly, staining with Congo red and gleaming with apple green birefringence under polarized light. Glycogen, too, may accumulate. In diabetes mellitus, hepatocytes, renal tubular cells, and cardiac myocytes brim with glycogen, demonstrable by PAS stain. In glycogen storage diseases, inherited enzyme defects produce massive accumulation, sometimes distorting the very architecture of organs. Then there are the pigments, nature's colors that also tell tales of disease. Some are exogenous. The black dust of carbon, inhaled in polluted air, collects within alveolar macrophages, later migrating to lymph nodes, a condition called anthracosis.
Tattoos are another benign example, pigment deliberately placed and sequestered by dermal macrophages. Endogenous pigments include lipofuscin, the wear and tear pigment, a yellow-brown residue of peroxidized lipids accumulating with age in heart and liver, silent testimony to the passage of time. Melanin, dark and protective, may be increased in certain conditions like Addison's disease. Hemosiderin, a golden pigment, represents stored iron. Local deposits appear after hemorrhage, as macrophages devour hemoglobin and convert it to hemosiderin. Systemic overload, whether from repeated transfusions or genetic hemochromatosis, may stain organs rusty brown and cause serious dysfunction. Beyond accumulation lies the harder fate of pathologic calcification. Here, calcium salts deposit in tissues where they do not belong, chalky and white, granular under the microscope. Two distinct patterns emerge. The first is dystrophic calcification, which occurs in dead or dying tissues despite normal serum calcium levels. Caseous necrosis in tuberculosis, fat necrosis in pancreatitis, atherosclerotic plaques, and damaged heart valves all become encrusting with calcium phosphate, sometimes hardening into bone-like structures. Grossly, they feel gritty, as though nature herself has fossilized the tissue. The second is metastatic calcification, which occurs in otherwise normal tissues but in the setting of hypercalcemia. Elevated calcium may arise from hyperparathyroidism, destruction of bone by malignancy, vitamin D intoxication, renal failure with secondary hyperparathyroidism, or sarcoidosis. Calcium preferentially deposits in gastric mucosa, kidneys, lungs, arteries, and pulmonary veins, sites that excrete acid and therefore harbor an alkaline microenvironment favoring precipitation. Though often clinically silent, metastatic calcification can impair organ function when widespread. Thus, intracellular accumulations and pathologic calcifications are not mere curiosities. They represent the ledger of a cell's transactions, its failed attempts at balance, its scars of injury, its marks of age. They are the residue of life under pressure, painted in fat, pigment, and mineral. And now, with accumulations described and calcifications traced, the chapter moves inexorably to its closing theme, cellular aging. For even without toxins, ischemia, or genetic errors, time alone is enough to erode cellular vitality. The story ends where all stories must, with age, decline, and the inexorable march toward death.
Section 10, Cellular Aging.
Aging is life's paradox. It begins at birth, yet its signs creep so slowly that they seem invisible, until suddenly they are undeniable. The hair whitens, the skin loosens, the heart stiffens, the brain falters. At the level of the organism, aging is familiar. At the level of the cell, it is both mysterious and inexorable. What then is cellular aging? It is the progressive decline in a cell's functional capacity and replicative lifespan, accompanied by molecular and structural damage. Unlike acute injury, it is not a single blow, but the gradual accumulation of countless small insults etched into DNA, proteins, and membranes over decades. One fundamental mechanism is replicative senescence. Normal somatic cells, unlike immortal cancer cells, can divide only a finite number of times. Each division trims the ends of chromosomes, the telomeres, repeating TTG-GG sequences that act as protective caps. With every replication, the telomeres shorten until one day they are too eroded to shield chromosomal ends. At this point, the cell perceives the unprotected DNA as damage and halts further division, entering a state of permanent arrest. This is senescence.
Germ cells, stem cells, and cancer cells evade this fate by expressing telomerase, the enzyme that rebuilds telomeric repeats. In most somatic cells, however, telomerase is absent or weak, and the countdown proceeds relentlessly, like a cellular clock. Another axis of aging is accumulated damage. The genome is constantly assaulted by reactive oxygen species, by replication errors, by environmental agents. Though repair mechanisms exist, they are not flawless. With time, mutations accumulate, proteins misfold, and organelles falter. Lipofuscin pigment collects. Mitochondria leak free radicals. Lysosomes weaken. It is a slow corrosion, not catastrophic but cumulative, undermining the vitality of the cell. Equally important is the deregulation of nutrient-sensing pathways. Cells, like organisms, have metabolic circuits that balance growth and conservation. Among these, the insulin and insulin-like growth factor, IGF-1, signaling pathway has emerged as a master regulator of longevity. In experimental models, dampening this pathway extends lifespan, while excessive activity accelerates aging. One downstream mediator is mTOR, a kinase that integrates nutrient signals and drives protein synthesis and growth. Its inhibition promotes autophagy, the recycling process that clears damaged organelles and proteins, thereby enhancing cellular survival. Thus, caloric restriction, by reducing IGF-1 and mTOR activity, extends life in multiple species. Here lies the fascinating suggestion that aging is not wholly inescapable, that its tempo may be modulated. At the tissue level, aging cells often enter a senescence-associated secretory phenotype. No longer dividing, they secrete inflammatory cytokines, growth factors, and proteases that alter the surrounding microenvironment. This creates a paradox. While senescence prevents malignant transformation of damaged cells, it also fosters chronic inflammation and tissue dysfunction, contributing to age-related disease. Ultimately, aging is the sum of these interwoven processes: telomere shortening, DNA and protein damage, oxidative stress, impaired autophagy, altered nutrient signaling, and senescence-associated inflammation. The result is diminished regenerative capacity, loss of physiological reserve, and increased susceptibility to disease. Clinically, aging manifests as frailty, impaired wound healing, neurodegeneration, sarcopenia, immunosenescence, and vulnerability to cancers. Yet it also manifests as wisdom and endurance. For aging, though biologically a decline, is the very canvas upon which the full picture of life is painted. And so the chapter closes. We began with the assaults of injury, traced the paths of necrosis and apoptosis, followed newer modes of death, dissected the hidden mechanisms, explored clinical correlations, observed the resilience of adaptation, cataloged the strange treasures of accumulation and calcification, and finally bowed to the inevitability of aging. The story of the cell is thus the story of existence itself: birth, adaptation, injury, death, and decline. To study pathology is not only to read of disease, but also to read, with scientific clarity and artistic awe, the very script of life.